Hi, in this session we will be discussing about Callus culture, and under Callus culture we will be discussing about morphology, internal structure and characters of Callus, and what all are the importance of Callus culture. So, coming to morphology of callus. Usually a callus tissue is an proliferating unorganized mass of cells, and these tissue or cells do not have any regular shape. So describing the external morphology of callus is difficult, but they can be distinguished based on certain other characters like texture, coloration or requirement of hormones, etc. And callus which are produced from different explants such as root, leaf, stem, flowers, etc. of the same species show considerable variations, and they can be identified based on the morphology. So as such, the callus doesn't have any regular shape, so describing a callus based on its morphology is very difficult. Second character of callus is the internal structures. So studies on the internal structure of callus has been done both by light and electron microscopy. And light microscopic studies, it has been found that the cellular composition of callus is extremely heterogeneous. There are different types of cells based on size, shape, etc. So within a callus we can see very small cells which are having dense cytoplasm, and also large cells which are having vacuolated cytoplasm. Not only the size, the shape also shows variation, and there are spherical cells to elongated cells which are present within the callus, and these small spherical cells are actively dividing cells and they have dense cytoplasm, and these elongated cells which are present within a callus are involved in vascular differentiation or cytodifferentiation. And by electron microscopic study we get a detailed idea about the internal structures which are present within a cell. Next character of callus is texture. Based on texture, callus can be divided into two, that is soft callus and hard callus. Soft callus are also called as friable callus. That means they are friable in nature. The cells of the callus can separate easily, and they are made up of heterogeneous mass of cells, and they have minimal contact with each other. That will be the reason why the cells of soft callus can separate very easily. Second division based on texture is hard callus, and hard callus usually consists of chain cells or trachea like cells, and these cells are closely packed, that is compact in nature. Next character of callus is the coloration. Usually callus has a creamish yellow or white in color, and in certain cases callus may be pigmented. As we had observed before, a callus which is in pale green in color can give rise to shoot, and a callus which is slightly yellow in color will give rise to root, we have seen before. So certain cases callus is pigmented, and this pigmentation within callus can be patchy, that is found in certain areas or certain group of cells, or their pigmentation may be uniformly distributed. And green color callus is formed due to development of chloroplast within the cells, and yellow coloration of callus is caused due to synthesis of carotenoid pigment. And in some cases culture of cauliflower lead to formation of callus which are purple in color, and this purple color callus is formed due to development of anthocyanin. And in certain plants they can form brown color callus. This brown coloration can also develop in the eggplant and also in the callus, and this brown color is developed due to excretion of certain phenolic substance. These phenolic substance may be present within the cells of the explant, and separated from these phenolic substance within the cells of an explant, there are also a group of enzymes which is called as polyphenol oxidase. And when we are cutting these explants, they the phenol and polyphenol oxidase come in contact, and this phenol gets oxidized to quinones by this particular enzymes, that is polyphenol oxidase, and this quinine oxidized to form the brown product. And these phenolic oxidases can act as a hindrance for the growth of callus. Next is habituation. As we know, a medium for callus initiation should be supplemented with both auxin and cytokinin. If we are culturing a callus in a medium for a long time, then it leads to a process known as habituation. Habituation means when callus tissue is grown subcultured for a large time, when we are growing this callus tissue on standard maintenance medium which is devoid of any hormones, then this callus is able to grow in that medium. This property is called as habituation. So prolonged subculturing of callus leads to a property where the callus is able to grow in a standard maintenance medium which is devoid of any hormones, and this property of callus is called as habituation, and such a callus is called as habituated callus. Next is the chromosomal variation in a callus. And chromosomal variation within a callus can occur genetically or epigenetically. Genetical variability are caused as a result of taking cells of the explant which has already undergone some genetic changes. So the explant which we have taken has already undergone some genetic changes. This can lead to chromosomal variation when this explant leads to formation of callus. That is called as genetic variability. And epigenetic variability means when we are culturing an explant in a medium, this may lead to some genetic changes. So genetic changes which are arising during culturing is called as epigenetic variation. And as we all know that subculturing leads to changes in ploidy level of the cells of the callus, and usually only diploid cells can give rise to callus or shoot or root, whereas the other polyploid cells cannot give rise to callus under tissue culture. These were all the properties of callus. Next, we are going to discuss what are the different significance of callus culture. So, the main significance of callus culture is that a whole plant can be regenerated from callus culture. So, from explant we produce a callus. From that callus first we initiate shoot, then we initiate root, and we can produce a whole plantlet from callus, and these plantlet can be produced in large number. Second property of callus culture is somatic embryogenesis can be initiated from callus culture. Third one is genetic and karyotypic variability can be produced from callus culture, and these genetic variable plants which are produced from callus culture may have some superior qualities, and they can be used for plant breeding experiments. Next, cell suspension cultures can be initiated from callus culture, and another significance of callus culture is that it can be used for obtaining useful secondary metabolites. Thank you.